Hello and welcome to today's JMCC ISHR webinar. Thank you for joining us for today's session. And thank you, as always, to JMCC and the ISHR for their continued support of our webinar series. So just a few quick announcements before we get started. Just a reminder, JMCC has started now 2 additional calls for papers of special interest. These will be open beginning from September to March. We have a call for papers for sarcomere protein mutations, mutations in iPS-derived models, and a number of other special topics, as you can see here. And we have a call for papers on the topic of ion homeostasis, calcium channels, and arrhythmias. So please check this out on the JMCC website if you are interested in submitting. Another reminder to check out the JMCC Scientist in the Spotlight podcast. There's 2 new episodes that have just gone online, including a really interesting interview with Lisa Kranius. So I recommend everyone to check this out as well. It can be found on a number of platforms or just search JMCC Scientist in the Spotlight. To save the dates, save the date for the ISHR North American Section 2022 meeting in Winnipeg from September 6th to 10th. You can see that here. The website will be launching shortly. And a reminder to save the date for the upcoming ISHR World Congress, which will be in Berlin from June 12th to 15th. So with that, I will say thank you again for joining us, and I will turn our session over to the chair of our webinar, Dr. Anja Karlstad, to introduce the speaker and get us started.
Thank you, Ron, and thank you everybody for joining us on this morning. It is my absolute pleasure to introduce today my esteemed colleague, Dr. Julia Ritterhoff. Julia is currently an instructor at the Mitochondrial and Metabolism Center at the University of Washington. She started her scientific career at the University of Heidelberg in Germany in 2013, where she received a doctorate degree in molecular and translational cardiology under tutelage of Patrick Most. She then joined Dr. Wang Tian's lab as a postdoctoral fellow at the University of Washington in 2015, where she studies how metabolic alterations drive the development of cardiovascular diseases. Julia has received multiple grants, including a postdoctoral fellowship award from the German Research Foundation, the ITHS Early Investigator Catalyst Award, and most recently an award from Agilent Technologies to support her research. Her studies focus on cardiac metabolism, and she and her work has advanced our understanding of how nutrients are metabolized in the failing heart. Just in the last year, Julia has published 2 first author papers, both in JMCC and Circulation Research, and I'm very glad that today she took time to talk to us about her very, very recent paper that was published in May in JMCC titled "Increasing Fatty Acid Oxidation Elicits a Sex-Dependent Response in Failing Mouse Hearts." Thank you, and let me welcome Julia again. Please take it away.
Thank you, Anja, for the kind introduction. I hope the screen sharing works. So if not, please let me know. Yeah, thank you for the kind introduction. I would also like to thank the editors from JMCC for selecting our paper and for giving me a chance to share our work in this forum. So today, as Anja alluded, I'll be talking about the sex differences in cardiac metabolism and the findings that we made when targeting fatty acid oxidation and heart failure. I always like to start with this EM picture from a normal mouse heart. As you can see, besides myofilaments, mitochondria are the second most abundant organelle in the heart, and they are tightly packed besides the myofilaments. Mitochondria is a so-called powerhouse of the cell and make most of the ATP. So this simple picture already highlights the importance of energy metabolism for cardiac function. And to give you an idea about how much ATP the heart actually needs, it's been estimated that the human heart turns over about 20 to 30 times of ATP its own weight, which is about 6 kilograms of ATP daily. So in order to meet this high energy demand, cardiac metabolism is flexible to use a variety of substrates for ATP generation. Under normal conditions, the heart mostly uses fatty acids, and glucose to a lesser extent. However, the heart is an omnivore, demonstrating high flexibility for any substrates in order to adapt to changes in workload or substrate availability. And for example, during exercise, increased availability of lactate or fatty acids in the blood shift the cardiac substrate metabolism towards these fuels. Given the importance of ATC synthesis for cardiac function, it's not surprising that metabolic remodeling occurs in heart failure. Under non-ischemic conditions, the heart switches from mainly using fatty acids to increased reliance on glucose and glycolysis for ATP generation, and in later stages also to an increased reliance on ketone bodies. Changes in substrate metabolism are among the earliest changes and actually precede contractile dysfunction. And while this switch might be compensatory to main energy production for cardiac function in the beginning, it becomes maladaptive and contributes to cardiac remodeling in the long term. Furthermore, reduced mitochondrial function coincides with decreased oxidative metabolism and results in decreased ATP synthesis. Sex differences occur in the healthy heart and in cardiovascular diseases. There are sex-dependent differences in age at which they become manifest, in the pathological consequences, and in response to treatment. Overall, female sex is a well-established protective factor, and female mice develop less severe heart failure under experimental conditions, such as pressure overload-induced hypertrophy. For example, while male mice respond predominantly with eccentric hypertrophy, severe fibrosis, and systolic dysfunction to pressure overload, female mice demonstrate a more concentric hypertrophy, moderate fibrosis, and preserved systolic function. Mechanisms for these differences have been partially explored and exclude genetics, epigenetics, and hormonal differences. Specifically, the role of hormonal regulation has been linked to sex differences in metabolic remodeling. In male hearts, heart failure is associated with a complex downregulation of transcriptional programs, such as the PPAR alpha and PTC1 alpha pathway. This includes downregulation of genes for fatty acid oxidation, oxidative phosphorylation, and lectin transport gene function. On the other hand, estrogen is known to regulate several of these processes. As a consequence, female mice demonstrate less inactivation of these pathways. There are many reasons why a heart can fail. A failing heart has been described as an engine out of fuel, where an imbalance between energy demand and supply results in contractile dysfunction. This is also highlighted by the observation that the energetic status of the heart, which is the PCR-ATP ratio, is a predictor of mortality in heart failure patients. However, I'd like to point out that this study was actually conducted mostly in male patients, where more than 80% of patients were male. So the importance of energy deficit for heart failure in females is actually little understood. Again, predominantly male studies in mice have proposed the concept of increasing ATP supply in the diseased heart. Comprehensive studies support the idea that increasing oxidation by providing sometimes supraphysiological substrate level can be beneficial to maintain energy production and prevent adverse remodeling. In this context, our lab has pioneered research on the beneficial role of fatty acid oxidation to prevent heart failure. And so the rate limiting step in fatty acid oxidation is the import of long-chain fatty acids across the mitochondrial membrane for GPT-1. This action is strongly inhibited by melanoid-CoA, which is formed by ACC2. Knocking out ACC2 inhibits the formation of melanoid-CoA and takes away the brake from mitochondrial fatty acid uptake, thus increasing fatty acid oxidation. And so to specifically target this pathway, our mice has taken advantage of an ACC2 knockout mouse, which shows increased fatty acid oxidation and a reduced glucose oxidation with no other notable effects on fatty acid or energy metabolism. And so in previous studies, we could show that ACC deletion in the heart maintains systolic and also diastolic function in different stress models. It also preserved cardiac energetics and attenuated hypertrophy. So these studies suggest that preventing the decline of fatty acid oxidation can mitigate the development of heart failure. And to explore the translational potential of this, we actually wanted to ask if stimulating fatty acid oxidation can be a therapeutic target in heart failure. And so we formulated the hypothesis that increasing fatty acid oxidation after the onset of pathological hypertrophy will attenuate heart failure progression or improve mitochondrial oxidation. Since there is no way to pharmacologically target cardiac ACC without systemic effects, we use our genetic models to delete ACC2 by tamoxifen injection. And to use a therapeutic approach, we aim to increase fatty acid oxidation during the transition from pathological hypertrophy to heart failure. To identify sex-specific mechanisms, we studied female and male mice in different cohorts. Additionally, Merck toxicity has been reported in tamoxifen-induced genetic mouse models, and so we use Merck mice as an additional control group to the regular flox wild-type mice, as indicated in this color key. For this first set of experiments, we ended up studying 3 groups of mice in both sexes. In a previous study, we could show that ACC2 deletion was as early as 2 weeks after tamoxifen injection, which resulted in a time-dependent increase in cardiac fatty acid oxidation. So the first confirmed that a similar time course was happening with tamoxifen injection after TAC. And as you can see, ACC protein was efficiently deleted in ACC2 knockout TAC mice 2 weeks after TAM injection, which should lead to increasing levels of fatty acid oxidation about 6 weeks after TAC. And so we first looked at cardiac function in both female and male mice over time. As you can see, TAC surgery induced a progressive decline in systolic function and also a continuous ventricular dilation in both control groups of either sex, which are depicted in black and gray. In male mice, ACC2 deletion did not change the time course of contractile dysfunction, which is the blue group. However, in female mice, systolic was improved, and also left ventricular dilation was reduced in ACC2 knockouts, which is the red group. So here, I'd just like to point out that besides having different time courses, fractional shortening was actually not much different between females and males at the end of the study in both control groups. However, only ACC2 deletion in females improved cardiac function. So we next looked at markers of hypertrophy and markers of ventricular remodeling in both sets of mice. In line with previous studies, pathological remodeling was less severe in females than males, is demonstrated by less hypertrophy and less increased expression of pathological markers. However, ACC2 deletion had no effect on hypertrophy or gene expression in either sex. So together, these first set of experiments show that ACC2 deletion differently affects cardiac function in female and male mice. Only female mice benefit from increasing fatty acid oxidation. With respect to our 2 control groups, the flox wild-type and the wild-type Merck mice, cardiac remodeling and function were not different, and so we only used 1 control group for the subsequent experiments. And so previous studies have shown that if we delete ACC2 before chronic stress, this can prevent mitochondrial dysfunction and metabolic remodeling. However, if this holds true, if the stress comes first, it's still known. Furthermore, if and how biological sex affects the response to mitochondrial and metabolic remodeling is unknown. So we first analyzed the expression of mitochondrial proteins in female and male hearts after TAC. We looked at electron transport chain subunits, as well as mitochondrial membrane and matrix proteins. And aligned with the literature, most of these proteins were actually downregulated in male hearts after TAC, were not affected in females. And similar to the before observed changes in molecular remodeling, mitochondrial remodeling was unaffected by ACC2 deletion in either sex. As pointed out before, metabolic remodeling is coordinated by a network of transcription factors and coactivators. In order to determine the upstream signaling responsible for sex-dependent metabolic remodeling, we turned our attention to the PGC1 alpha signaling complex. PGC1 alpha expression itself was unaltered in male heart, but the expression of several interacting factors such as NRF1 and 2, PPAR alpha and RXR alpha were downregulated in both control tag and ACC2 knockout tag. PGC1 alpha expression was also unaltered in female ester tag, as was the expression of the other transcription factors. Surprisingly, expression of PPAR alpha and RXR alpha was actually higher in female ACC2 knockouts after tag. We then looked at target genes for mitochondrial biogenesis, which were downregulated in both male control tag and ACC2 knockout tag. In contrast, these were unaffected in female tag hearts. In line with this, mitochondrial DNA content was also reduced in male, but not in female hearts. Target genes for mitochondrial fatty acid oxidation were also then regulated in male hearts after tag, and even further reduced in ACC2 knockouts. In contrast, gene expression was unaltered in female control and ACC2 knockout tags. Collectively, these data show that tag down-regulated pathways for mitochondrial function, biogenesis, and fatty acid oxidation in male were not in female mouse hearts. These changes were not reverted by targeting ACC2. While these data suggest that the oxidative capacity was preserved in female mice after tag, it did not explain why ACC2 deletion improved cardiac function. So we decided to perform another set of experiments. Considering that fatty acids could be a more effective supply for energy during increased demand, we next tested if increasing fatty acid oxidation after tag improved cardiac energetics. And in order to determine the early effect of ACC2 deletion, these experiments were performed when cardiac function was similar in control and ACC2 knockout mice at 6 weeks post-tag. And so using a combination of isolated perfused heart experiments, 31P NMR and 30C spectroscopy, we determined how changes in substrate supply affected the energetic status of the heart in response to higher demand. And so I just want to give you an idea how these experiments were performed, since they're quite technically challenging. And so we first performed Langendorff perfusion, where you can see that the heart actually beats, or maybe, yeah, now you can see it, hopefully. And then combine this with 31P NMR spectroscopy, where you have to stick the beating heart into a small tube and actually fit this in a big magnet, which allows to simultaneously assess contractile performance and energetics. And to further complicate these experiments, we use the high calcium stimulation to stimulate higher contractile performance. And so for these experiments, we actually also included a group of sham hearts to monitor the effects of tags in both female and male hearts. As you can see on the left side, high workload increased the developed pressure in all group of hearts in male and females. However, male hearts after tag were unable to maintain the heart rate during this high workload period. And this, as a consequence, control and ACC2 knockout tag hearts failed to increase the rate pressure product. In contrast, female sham and tag hearts, heart rate was maintained, and the RPP similarly increased in all groups. And so on the left, you can see a classical 31P spectra that allows you to calculate the PCR-ATP ratio, which represents the energetic status of the heart. And so we monitored this during the high workload period. And as you can see in male heart, this resulted in a moderate decline in the PCR-ATP ratio in L groups. In contrast, in female hearts, only the PCR-ATP ratio only dropped in the control TAC group, but not in sham and ACC2 knockouts. And so together, this suggests that the depletion of the energy reserve presented an increase in cardiac performance in male hearts, whereas in females have maintained cardiac performance at the cost of the energy reserve. So this difference becomes more clear when looking at the relation of the RPP, which is the indicator of the cardiac work, and the PCR-ATP ratio. And as you can see in males, TAC induces a left shift, suggesting that male hearts maintain the cardiac energetics by limiting cardiac work. On the other hand, in female hearts, TAC demonstrates the downward shift, suggesting that they mobilize their energy reserve to maintain left ventricular function. Importantly, ACC2 deletion had a different effect here in both sexes. The ability to maintain cardiac work in female hearts was associated with sustained cardiac energetics, while no improvement was seen in male hearts. And so together, these results suggest that metabolic remodeling during heart failure limits the energy supply in TAC hearts, whereas only female hearts benefit from increased fatty acid oxidation. And so finally, we also looked at the substrate utilization using 13C NMR in the hearts. And if you're familiar with these kinds of experiments, you can tell by just looking at the glutamate peak that the substrate utilization is different in control and ACC2 knockout hearts after TAC. And quantitative analysis confirmed that ACC2 deletion increased fatty acid oxidation in isolated hearts during the high work challenge in both sexes. So if fatty acid oxidation is increased in both males and females, why doesn't that improve cardiac function and energetics in the males? As demonstrated so far, heart failure in the male mouse heart is associated with a complex downregulation of metabolic pathways, including expression of genes in fatty acid oxidation, TCA, and energy metabolisms. So what happens if we actually push more fatty acids into these dysfunctional mitochondria when we delete ACC2? So we speculated that increasing mitochondrial fatty acid import in the face of reduced oxidative metabolism would lead to incomplete fatty acids, incomplete oxidation of fatty acids in male hearts, thus limiting the effectiveness of energy production. And to test this, we turned our attention to the acetyl-carnitine, which are an intermediate of mitochondrial fatty acid oxidation and serve as a marker of incomplete oxidation. And so we observed significant increase in total acetyl-carnitines in the male ACC2 knockouts heart after TAC, which was much more moderate in the female. And so we looked at the whole list of different acetyl-carnitine species and found that several medium and long-chain acetyl-carnitines were increased in male ACC2 knockout after TAC, but not hydroxylated. And overall, the acetyl-carnitine profile was more variable in females. No statistically significant was present. And additionally, we found that some of the long-chain acetyl-carnitines inversely correlated with cardiac function in both male and female ACC2 knockout mice after TAC. And this further supports our conclusion that incomplete fatty acid oxidation caused by impaired mitochondrial oxidation contributes to the differential response to ACC2 deletion in female and male hearts. And so one important note here is that acetyl-carnitine levels were not different between control and ACC2 knockout mice of either sex under unstressed conditions. So it is likely that the accumulation of acetyl-carnitines in TAC was a consequence of reduced mitochondrial oxidative capacity and not a consequence of enhanced mitochondrial fatty acid import. So how do we fit all together? A healthy heart is able to efficiently oxidize fatty acids to meet its energy demand. However, chronic stress, such as TAC, reduces mitochondrial oxidative capacity in male hearts. And this limits the ability to upregulate fatty acid flux to match the increased import of fatty acids after ACC deletion. This creates an oxidative bottleneck and results in accumulation of acyl-carnitines. In contrast, in the female heart, oxidative capacity is sustained after TAC. Here, increased import of fatty acids into the mitochondria is adequately oxidized, which ultimately improves myocardial energetics and cardiac contraction. And so, despite answering some of our initial questions, I feel I actually ended up with more at the end of the study. For example, elevated levels of long-chain acyl-carnitines are associated with adverse outcome of heart failure patients and can directly contribute to cardiac dysfunction. However, in our study, pathological remodeling and cardiac dysfunction was not worse in male ACC2 knockouts after TAC, despite the accumulation of acyl-carnitines. So one question that remains is if acyl-carnitine have actually detrimental effects in our study. And next, our genetic approach limited the timing of our therapy. It still remains unknown if an earlier increase in male mice could improve the outcome, whereas a later increase in fatty acid oxidation in female mice could actually prevent beneficial effects. And on a more general line, while we investigated the response to our therapy in female and male mice separately, how fatty acid oxidation is regulated in female hearts and under failure still remains poorly understood. And looking at the translational perspective, in combination with stimulating substrate metabolism, can we find targets to stimulate oxidative capacity, specifically in the male heart for therapy? So these are just a few questions to think about. So before closing, I'd like to thank the folks in the Cell Lab who contributed to this story, specifically Ron, who supported me throughout the way, and Tim, who's a very talented staff scientist and did all the perfused heart experiments. Then I'd like to thank the mass spectrometry core for helping to develop our metabolomics assays, and of course, the JMCC for early faculty support. And thank you all for zooming in, and I'm happy to take questions.
Well, thank you so much, Julia, for this very intriguing talk. And just a reminder to everybody, please, if you have questions, insert them into the Q&A section and we will then read them individually. But so before everybody gets started, let me take off with some questions. You were talking about the differences in the way fatty acids are oxidized between males and females, and I was wondering if you noticed a difference in the chain length of fatty acids that are utilized. Is there an increased uptake of long chain or medium chain fatty acids, and is there also a sex difference between your groups?
That's a good point. So I think the general concept is that female have a higher fatty acid metabolism at baseline. And actually in our studies, we're not able to discriminate between fatty acids. So kind of the cardiac functional studies were performed in regular mice when they eat their normal food. So we actually didn't look at specific fatty acids. And for the perfused heart experiments, we only use long chain fatty acids. And these are all labeled. So for the 13C tracing experiment, we cannot discriminate between different saturated or unsaturated fatty acids.
But they were basically in the C16 and above length, or did you?
Yeah, so they're all, no, so they're kind of classically long chain, C16, 18, and then saturated and unsaturated ones.
And before everybody gets ready with their questions, I was wondering if basically your concept that you're showing is when I was looking at the data and reading your paper is that in a way females are mobilizing and storing the nutrient reserves in a very different way than males. And in the context of stress or physiological or mutations, they are then able to kind of mobilize them in a more efficient way. Is that a correct assessment of your study?
Yes, I think we didn't look that much at how they store energy with respect, for example, to triglycerides. I think we looked at that and didn't really see a difference under any conditions. I think we think about it more in a way how they use incoming nutrients, and that for males we understand way better how they use nutrients, specifically fatty acids, and that in heart failure, you have quite dysfunctional metabolism and mitochondrial dysfunction, and so they can't really use it. And I think what we see in our study is that the female hearts, they can still use it. And if we stimulate that pathway, they actually do better after stress.
So there is a question from Bruno Muquet. He writes, thanks for your presentation. Does phosphorylated ACC2 have any impact on the different mouse models you used in your strategy, and also fatty acid oxidation?
So we looked at phosphorylated ACC2, but we actually did not see any changes. I mean, since we deleted in our knockout strategy, it actually is not phosphorylated anymore in other conditions. But I don't think we haven't compared directly between female and male if there would be a change in phosphorylation. So that might affect fatty acid oxidation.
Okay. Then there is another question from Asan. Excellent presentation. I wanted to know if you find differences in glucose or ketone metabolism between males and females in the heart failure model.
And so the way we assess substrate metabolism is we can measure the relative contribution from different substrate in the perfused heart. And then when we see an increase in fatty acid oxidation, that resulted in a decrease in glucose contribution. These experiments were not performed in the presence of ketone bodies. And also we don't assess absolute oxidation, so direct comparison is actually difficult to make.
Okay. Then Sarah from Madrid would like to know if you also checked for mitochondrial respiration differences between males and females before and after TAC, and what was the age of the male animals for the TAC, and do you think there's a change in or differences that age might play? So mitochondrial function and then age.
Yeah, so unfortunately we did not assess mitochondrial function in isolated mitochondria, and I think at the end of the study we just didn't have the live material to do this, and I honestly would be very curious to look at that as a functional readout, because I think what we see is that gene expression does not always translate in functional outcomes. So maybe we'll do that in the future. The second part, the age of the mice. So the male mice were actually around 10 weeks, whereas the female mice were a little bit older, 13 weeks. We actually got this question from a reviewer during publication and asked if that could affect the outcome. But honestly, if you look across literature, pathological studies are performed in mice between 8 to 12, 13, 15 weeks. And so also we in our lab have done studies looking at mice in that range and have not seen any differences in heart failure phenotype. or metabolic remodeling. So yes, the mice had a different age, but I don't think, based on our previous observation, that this affected our outcome. It was more a practical approach because, as you might know, female mice are smaller. So if we performed the TAC surgery at the same age, we actually had a different pressure overload. So we waited a little bit to get them a little bit bigger.
Great. Let me ask you also, when it comes to the fatty acid metabolism, of course, one like downstream effect would be fatty acid oxidation, but also lipid remodeling in general might play a role in your model. Do you plan on doing any experiments regarding assessing how maybe the phospholipid metabolism is altered in the model? Are there differences that you are aware of? And have you guys come up with a hypothesis regarding this?
Yeah, so of course, lipid remodeling is important, and also for heart failure. So we've done some studies in the ACC2 gut mice just under regular or baseline conditions, and haven't seen any changes in TAC or TAC content. And so we tried to look at that in this model as well, but actually couldn't see any changes as well. So I think it would be interesting, for example, to look at the phospholipids maybe in the mitochondria. So these studies are definitely interested in maybe something to look at in the future.
Yeah, we just got another question from Helen Collins. She would like to know what was the stage of estrus that the mice were in, the female mice, and also do you think your sex-dependent findings regarding the lipid metabolism are also the same in a model of myocardial infarction?
These are tough questions. So unfortunately, we did not look at the estrus or the cycle in the female mice. And then the second part, let me take out that question.
Is the model also applicable to myocardial infarction? So can you, instead of using TAC, would you expect a similar change between males and females?
Yeah, I mean, I think metabolic remodeling after general cardiac remodeling after MI is quite different than during chronic pressure overloads. If you have an MI, you have the acute generation of myocardium, whereas during pressure overload, you have the primarily hypertrophy response. So we haven't performed these experiments in an MI or ischemic model. I mean, in general, sex-dependent differences in ischemic and SALT, I think, is just as relevant, as important as during pressure overload. So something to do in the future.
Well, Mathieu is asking, and so there's increasing work. He points out there's increasing work showing that peroxisomes are also potentially compensating for some of the mitochondrial failure and to correctly use fatty acids. Is that something that you have been thinking about? Have you done experiments in that area?
Yeah, so we were kind of thinking along that line at one point, if the peroxisomes could perform some kind of fatty acid oxidation or lipid oxidation. We kind of looked a little bit at gene expression and actually could not find any difference or any effect of TAC or SAX, but we didn't explore this to the full extent.
Mm-hmm. Do you think that the differential peroxisome mitochondrial fatty acid metabolism might play a role in that model that also explains the differences in males and females in response to TAC?
I mean, I guess at this point I can't exclude it, but I also don't have any experimental evidence for that.
Well, thank you, Mathieu, for the suggestion.
Yeah.
And you were tackling this from a fatty acid oxidation point of view, but we all know that fatty acids can be taken up, they can be synthesized. Do you think there are other targets in this pathway that would be similar metabolic liabilities that we could target pharmacologically, in addition to targeting fatty acid oxidation directly.
Yeah, I think that's what I tried to point out in the end a little bit, that the differential response we see in females and males, and especially the reduced oxidative capacity in the males, is probably a good target either alone or in combination with targeting substrate metabolism. So something to stimulate mitochondria directly and general oxidation.
Well, I guess those were all the questions as far as I can see. Thank you, everybody, for this very intriguing discussion. And thank you very much, Julia, for your time and presenting your work. I hope we can re-invite you in a couple of months or next year with your follow-up studies. So thank you very much. And thank you, everybody, for attending today's webinar. Have a wonderful day and rest of the week. And yes, Melissa, the webinar is being recorded, and Ron just mentioned to me later this week, a recorded video will be posted on YouTube. So we will announce this also through the website, Twitter, and the other messaging systems from JMCC. So you can see the video and Julia's presentation on YouTube. Thank you. Thank you, Julia.